Hi, in this video I will talk about urea cycle and why urea cycle is important for our body. So urea cycle is one of the most important biochemical pathways in our body because in our body when protein is broken down it forms amino acid, and when amino acid is broken down ammonia is produced, and ammonia is highly soluble in water, and our body is mainly composed of blood, which is actually more than 90% water. So ammonia could get solubilized in the blood and can increase the level of toxicity. That is why our body has to devise a mechanism by which it can eliminate the ammonia which is produced from the amino acid catabolism. And urea cycle is that biosynthetic pathway which ensures ammonia is eliminated properly and our body is safe from ammonia toxicity. So the reaction of the urea cycle is happening in the liver and to some extent in kidney. So here is the liver, and if we zoom into the liver, so you can see there are lobules, hepatic lobules, hexagonal hepatic lobules composed of hepatocytes. Hepatocytes are liver cells. So if we zoom, this is the hepatocyte, one liver cell. So all the reactions of the urea cycle is taking place in the mitochondria and the cytosol. Some portion of the reactions are in the mitochondria and some portion of the reactions are taking place in the cytosol. So let's just zoom into the mitochondria and see the reactions in step by step. So here we have the mitochondria of the hepatocyte. So what happens, all the amino acids which are broken down are actually funneled in form of glutamate. By the reaction transamination, they are usually formed glutamate. And glutamate would ultimately get converted into ammonium ion. And on the other hand side, glutamine, which entered the mitochondria, can be converted into glutamate by glutaminase, mitochondrial enzyme glutaminase. So the first step of the urea cycle is combination of bicarbonate and ammonium ion, and this is triggered by carbamoyl phosphate synthetase or CPS1 enzyme, which ultimately forms the first product, which is carbamoyl phosphate. And now the ornithine, which is present in the mitochondria, is actually combining with carbamoyl phosphate forms citrulline, and citrulline would leave mitochondria. In the cytosol, citrulline would form citrulyl AMP intermediate with the ATP present in the cytosol. Then aspartate would get combined with citrulyl AMP and it would form aspartate arginosuccinate. Now from aspartate arginosuccinate, arginine would be produced and fumarate would be released. Now this arginine would regenerate ornithine. So in this cycle I am skipping all the details of the enzymes, but I have mentioned the rate limiting enzyme, which is carbamoyl phosphate synthetase. So arginine would be ultimately converted into ornithine, and ornithine would again perform this cycle by getting converted into citrulline, and the whole cycle would be repeated. So from arginine to ornithine conversion step, as a byproduct, urea is formed, and urea is less harmful than ammonia. So we started with ammonium ion and we ended up with urea, and urea is excreted by urine. So this is how harmful ammonia is actually excreted out by urea by the urine as a form of urea. So that is why we are uricotelic. So excretion product of human is urea. So here is the regulation of urea cycle. So there are twofold regulation. One is genetic regulation, another is allosteric regulation. So genetic regulation includes expression of the urea cycle enzymes. People that are taking a diet full of protein have greater expression of urea cycle enzymes. However, In the starving condition, the carbon skeleton of glucogenic amino acids are broken down in order to form energy, and side by side ammonia level increase. In order to decrease that ammonia level, urea cycle enzymes are overexpressed in this situation. Allosteric regulation involves regulation of CPS1, that means carbamoyl phosphate synthetase 1, which is allosterically regulated by N-acetyl glutamate. N-acetyl glutamate would actually positively regulate the urea cycle. And on the other hand side, N-acetyl glutamate is produced from glutamate and acetyl-CoA, and arginine is acting as a positive regulator of N-acetyl glutamate synthetase, which is actually forming N-acetyl glutamate. This is how arginine and N-acetyl glutamate is positively regulating the reactions of urea cycle. I hope you enjoyed the video. If you like my video, please like and then subscribe. Thank you.